Great. Well, thanks so much to the organizers for the invitation. I'm thrilled to be here and tell you about some of the work going on in my lab to understand circuit mechanisms of value-based decision-making. So one of the main questions that my lab is interested in is how our subjective value is impacted by context. So just to give a really explicit example, this is a picture of people waiting in line for a restaurant in New York City, and how long they're willing to wait, and accordingly how much they value the restaurant is going to be strongly sensitive to the local context. So if the restaurant is in the West Village, which is where the NYU's campus is, this is a very restaurant-dense neighborhood, right? And so you might be willing to wait less time because you could just go next door and find an equally good meal. In contrast, if you find yourself in a more restaurant-sparse neighborhood, so this is deep in Greenpoint, Brooklyn, you might be willing to wait longer because it's going to be more challenging to find something nearby. So figuring out the solution to this problem, how long do I wait for the restaurant, is fairly straightforward when you have Google Maps on your phone and the environment is what's called fully observable. But my lab is interested in the more complex scenario where the environment is only partially observable and needs to be inferred based on ambiguous sensory evidence. And so that forms the inspiration for the behavioral task that my lab developed for rats. So in this task, rats initiate a trial by poking their nose in a central nose port. And when they do that, they hear a tone. And so the frequency of the tone tells the rat how much water reward it's being offered on that trial. There's 5 distinct tones that uniquely map onto 5 different volumes of water. So 5, 10, 20, 40, or 80 microliters. Then one of the side ports lights up, and that tells the rat, okay, the reward's coming at this location, but it comes with an unpredictable delay that's drawn from an exponential distribution, and on some fraction of trials, it never comes at all. And so the decision that the animal has to make is, do I continue to wait for this uncertain reward that may or may not come, or should I opt out of the trial by poking in the unlit port? And then the rat has the opportunity to start a new trial. So here's a video of one of our rats doing this task. On this trial, he hears the 5 microliter tone, and the reward port on this trial happens to be on the right, but it's a catch trial, so the reward won't come. So he ends up opting out at about 6.5 seconds. This is the same rat, this time on an 80 microliter trial, and the reward port's on the other side on this trial. So 80 microliters is a lot of water for a rat. So you can see he's really perseverating in the poke. But this is also a catch trial, so he opts out at about 11 seconds. So one of the things that I like about this task is that in contrast to standard 2 alternative forced choice tasks, where you have a binary behavioral readout, right? Choose left or choose right. Here we have a continuous analog behavioral readout of the animal's latent subjective value of the reward, which is how long is he willing to wait for it. So my lab has developed a high-throughput behavioral training facility to train rats in this task. So we built all of these training boxes. This is custom hardware that we fabricated. So inside each of these boxes is one of those behavior rigs with a touchscreen monitor. And we also set up this software infrastructure. So each of the rigs is automatically pulling version-controlled software from a Git repo and writing data to a MySQL database. It knows which rat is supposed to run based on the database and automatically pulls that rat's parameters from the previous day and writes new parameters at the end of the day. And what this allows us to do is parallelize and automate in software the training of about 100 animals per day. So the goal of the facility, in my view, is really threefold. The first is to generate large pools of trained subjects for experiments to mitigate the main bottleneck in behavioral neuroscience, which is actually training the animals to do interesting tasks. Second, the facility generates statistically powerful data sets for behavioral modeling, which is a key approach that my lab uses. And finally, we're interested in leveraging individual variability as a feature that's revealing of neural correlates as opposed to a bug to be averaged out. And so here I'm showing you data from 470 rats that have been trained on our task. It's difficult to visualize the behavior of so many animals, so I'm just showing you the average, but we're very interested in really digging into individual variability. And I should say I'm going to show you a lot of the behavioral characterization of how animals are solving this task, and that's the thesis work of a former graduate student, Andrew Ma. Okay, so to look at context-dependent subjective value, we then expose the rats to these hidden states or blocks of trials that differ in the distribution of rewards that they offer. So in what we call mixed blocks, rats can see any of the 5 reward offers. In low blocks, they only see the smallest 3, and in high blocks, they see the largest 3. And this one reward, 20 microliters, is present in all of the blocks, so there's always some ambiguity about the state that the animal's in. Nonetheless, we find that with a lot of experience and training, the rats become sensitive to these hidden states. So this is an example rat that's similar to the restaurant example. When he's in a very rich environment like the West Village, he's waiting less time for the same reward, 20 microliters, compared to the green point homologue, which would be the low block. Okay? And across hundreds of animals, we see on average about a 10% reduction in how long they're willing to wait for 20 microliters in a high block compared to a low block. So 10% is a comparatively large contextual effect in the literature. Okay, so we're really interested in understanding how the animals solve this task, and so we developed a psychological process model that was initially inspired by normative foraging theories, and specifically the marginal value theorem. So the idea is that what the rats should be doing in this task, in principle, is waiting until the value of waiting falls below what's called the opportunity cost, which in the optimal case would be the long-run average reward in the environment. And so the idea is that in a high block, the opportunity cost is high because the longer I wait, the more I might be missing out on 80 microliters on the next trial. Whereas in a low block, the opportunity cost is low because the longer I wait, I'm not really going to get a much better offer. And so this framework both provides kind of a normative foundation and just a useful qualitative description for what might yield shorter wait times in high blocks and longer wait times in low blocks. However, the question remained, how exactly did we think the animals were estimating the opportunity cost? And here I just want to kind of circle back to one of the take-homes of Eve Marder's talk, which is about the degeneracy of readouts. So one thing that we worry about a lot in my lab is the fact that behavior is degenerate, right? You could have a number of different cognitive strategies that give you the same behavioral readout. And I think it's really important to try to convince yourself that you have a reasonable understanding of the underlying algorithmic computations that are producing those behavioral readouts. So one of the things that my lab has tried to do to gain traction on this is really look at behavior in as many different ways as possible. So we use rich, multifaceted behavioral readouts, and the hope is that the more ways that we look at behavior, the better we can adjudicate between competing psychological process models. And so this has been an ongoing effort in my lab for several years, and I'm just going to show a couple of examples to try to give a sense of how we try to approach this problem. But to basically summarize over many of these studies, our current view, based on the data, is that the really well-trained rats that have experienced the blocks for months have basically learned about the latent block structure, and what they do is they infer the most likely block and then use a fixed block-specific estimate of opportunity cost based on that state inference. Okay, and so we model that inference process using Bayes' rule, where they're computing the posterior belief of a block given a particular reward offer by combining the likelihood of seeing that reward given that they're in that block with their prior belief about the block. Okay, and so this model captures a number of features of the rat's behavior, including the dynamics by which the wait times change at block transitions, also the fact that the wait times converge to kind of a common value in mixed blocks is consistent with them using a fixed block-specific estimate of opportunity cost. We can also fit the model using maximum likelihood methods, and it outperforms competing process models. The model predicts how the rat's behavior changes as we vary a number of task parameters. So for instance, we can play around with the probability that the trial will be rewarded or not, and we find that the rats adjust their wait times in a way that's qualitatively consistent with what the model predicts. We also find that the rats' wait times for the same offer, let's say 20 microliters in a mixed block, is not sensitive to the volume offered on the previous trial. And that's because we think the rats are using within a block a fixed block-specific estimate of opportunity cost. So that's present both in the data and is well captured by the model. And another thing that the model captures are kind of rapid inferred state transitions at highly informative trials called incongruent trials. So one of the features of the task, because of the way the reward distributions are set up, is if you transition from a low block into a mixed block, for instance, the first time you encounter 40 or 80, that tells you unambiguously that the world has changed because those rewards were not present in the previous block. Okay, so we call those trials incongruent trials. And those should happen pretty quickly. And so consistent with rapid changes in inferred states, if we break up the mixed blocks into quartiles, we find that the rats dramatically adjust their behavior within the first quartile, and then it remains stable. And moreover, we can look within the first quartile and find the trials after the first incongruent trial. And what we find is that if we look at the wait times conditioned on the previous block, it doesn't matter what the previous block was. They basically converge to a common value following these highly informative trials that indicate that the world has changed, okay? And importantly, these qualitative features are also captured by the model. So I'm just giving you a sense of kind of the range of behavioral readouts that we use to adjudicate between competing process models and convince ourselves that we have a decent handle on the underlying computations. And so based on that exercise, we think that when the rats are deciding how long to wait for uncertain rewards, they're performing this hidden state inference computation. And I don't have time to get into it today, but I just want to mention, to this point about degeneracy, we also find that when rats initiate trials, their trial initiation times are also sensitive to the blocks, but they reflect a fundamentally distinct computation, which is a standard delta rule or a reinforcement learning rule, okay? And if we were to just look at a very coarse level, we wouldn't be able to adjudicate between these. But using all of these higher dimensional readouts of behavior, it's very obvious that there's different underlying computations. And what we found over a series of studies is that these sequential actions are also supported by dissociable neural circuits. And so I'm happy to talk about the dopamine striatum work offline if anyone's interested, but for the sake of today, I'm going to focus on the role of orbitofrontal cortex in state inference. Okay, so the OFC work is largely the work of a former graduate student, Shannon Sherrick. And so one of the first things she did in the lab was bilaterally inactivate the orbitofrontal cortex using mucimol, which is a GABA agonist. So this basically, when you infuse it in brain tissue, shuts off all of the spiking in that brain area. And what she finds is that we see what I think is a kind of interesting effect, which is a reduction in the contextual modulation of the wait times. So if we look at control sessions, if you focus on 20 microliters, for instance, the rats strongly adjust how long they wait for 20 in a high compared to a low block. But when we inactivate the OFC, that sensitivity to the blocks is reduced, although it doesn't completely go away. We can look at higher resolution and how the wait times change at the block transitions. And what we find is that eventually the mucimol rats do adjust and modulate their wait times based on the high and low blocks if you look at the end of the block. But it just seems like it takes them longer to get there. And we can quantify that by breaking up the high and the low blocks into early and late trials. Okay? And so in the control cases, even in the early trials, the rats' wait times are sensitive and discriminating between the high and low blocks. But when we inactivate OFC, it really takes them to the end of the block to get there. So we wanted to use our psychological process model to articulate a more precise hypothesis about the underlying computation that was being disrupted here. So again, the model uses Bayes' rule to compute a posterior belief about the current block. And the way that Bayes' rule works is that the posterior on one trial becomes the prior on the next trial, right? So you have this recursive updating of beliefs over trials. So what Shannon found is that if she makes that recursion stochastic, such that there's some probability with which your prior just stays the same as it was on the previous trial, so you have like a sticky prior, she found that this was able to recapitulate all of the effects that we saw with our mucimol inactivations. So we see a reduction, but not an elimination of the contextual sensitivity and a slowing down of the wait time dynamics at these block transitions. So this gives us a fairly precise hypothesis about the computation that the tissue is performing, which is updating subjective beliefs based on outcomes. So based on that, we'd like to identify neural correlates of that computation. So Shannon performed recordings from 35 rats implanted chronically with neuropixels probes. And I'm just going to show you kind of two main pieces of electrophysiological evidence, but because this is a very complex, rich data set. So obviously, she found neurons in the OFC that are sensitive to the block. But one thing that we wanted to kind of differentiate was sensitivity to the block to sensitivity to the reward offers, because we know the reward offers are correlated with the block, and one of the things that OFC cares about is value. So what Shannon decided to look at was trials only offering 20 microliters. So the reward offer is the same on these trials. And what we're going to compare are trials offering 20 microliters in high or low blocks before the first incongruent trial into a transition in a mixed block versus after. And so we're going to first separate cells based on their preferred and non-preferred block. And our hypothesis is that if these neurons are reflecting inferred state transitions, then before the first incongruent trial, the firing rates of neurons should be different because we're selecting them based on having a different firing rate for preferred and non-preferred blocks. But critically, after the first incongruent trial kind of tells them that the world has changed, then we should see the firing rates converge to a common value because they should have inferred that they're now in a common mixed block. And again, I want to emphasize that we're only comparing trials offering 20 microliters only, so we're effectively marginalizing out the influence of block. And so that's exactly what we see. So this is showing neurons recorded from expert rats in the orbitofrontal cortex at the different events in the task. And so before the first incongruent trial, the firing rates are different for 20 microliter offers in preferred versus non-preferred blocks. But after this first incongruent trial, we see that the responses basically collapse onto each other. So I want to say two things about this. The first is that the incongruent trials are rare, right? We only get maybe 1 or 2 per recording session. And so we basically just took a brute force approach in terms of number of rats and number of neurons. The second piece I want to see is that this is frontal cortex, and I think it's pretty remarkable that we see such an interpretable signal of a potential cognitive computation at the level of single neuron firing rates. Okay, but we also wanted to analyze a feature of the population dynamics, especially because we have recordings from thousands of neurons. So this is the work of a very talented graduate student, Danilo, who's jointly advised by myself and Christina Savin. And what Danilo developed was basically a hierarchical linear dynamical system that could account for the observations or spikes of simultaneously recorded neurons as reflecting lower dimensional latent dynamics that explicitly vary over multiple timescales. Okay, so the idea is that the spikes or observations reflect a set of low-dimensional latent factors. These intermediate or within trial factors Y you can think of as kind of principal components that reflect time-varying dynamics within a trial. But the thing that separates this model from a standard Kalman filter is that those intermediate latents are themselves driven by longer timescale latents that we call Z, where Z is basically updated on each trial. Okay, and so the idea is that Z is impacting your fast latents, which is then impacting the spikes. And so we're explicitly partitioning the data into slow timescale and fast timescale components. And we can do things like show that if we remove one of the neurons, we could still predict its activity because that neuron doesn't exist in a vacuum, but is reflecting these shared low-dimensional latent dynamics. So one of the cool things about the model is that it's fully unsupervised, meaning that it only is fit to the spikes of simultaneously recorded neurons. It doesn't know anything about the behavioral task. And yet, the fast intermediate latents Y end up learning both the timing of salient task events and the within-trial variables that are important for the computations. So this is showing for one recording session the top 4 fast latents that explain the most variance in the data. And you can see that so we've aligned them to the time of reward, and the model has learned the timing of the reward and also has learned the reward offer on each trial. And over many sessions, we can decode the offered reward from these fast latents significantly above chance. Okay, so that's the within-trial latents. What about the across-trial latents? So those nicely reflect the inferred block, and that's true for one example session and also across experts. And so similar to what we did with the single neuron firing rates, we can also align these across-trial latents to these incongruent trials that are highly informative of state transitions. And in the expert rats, we see a clear separation before the transition and then collapsing to a common value afterwards. Whereas if we record from naive rats as they experience blocks for the first time, we don't see similar dynamics. Okay, so far I've shown you that OFC inactivations impair belief updating. Single neurons and latent neural factors estimated from population dynamics reflect these inferred state transitions. I didn't show you this, but it's in the preprint. We don't see similar correlates in other brain areas like M1 piriform or V2. So everything is not everywhere. This seems to be specific to this part of the brain that we've shown causally supports a computation for state inference. And we don't see similar correlates in rats early in training when they use a different strategy called divisive normalization, which I'm happy to talk about offline. Okay, so that was kind of cool, but a little bit unsatisfying because we'd really like to understand how at a circuit level this computation is being instantiated. So we have a number of projects that are ongoing in the lab now trying to dig deeper into how projections from OFC and projections to OFC from other brain areas support this belief updating computation. And today I'm just going to highlight one of those projects, which is from a super talented postdoc in the lab, Maggie DeMade. And she has been looking at the role of orbitofrontal cortex projections to the striatum. Okay, so why the striatum? So the striatum is the input structure of the basal ganglia, and in reinforcement learning accounts of the basal ganglia, it's demarcated into a ventral striatum, which includes the nucleus accumbens, which is thought of as a critic that's learning kind of the value of different states. And by virtue of the ventral striatum's projections back to the midbrain dopamine neurons, which instantiate error signals for learning, the critic is thought to be training a dorsal actor to select actions that maximize future expected rewards, okay? And so the OFC is known to project to both the dorsal and the ventral striatum. And so one of our first questions was, are those different neurons that project to those different parts of the striatum? So we did a series of anatomy studies where we injected different retrograde tracers that were labeled to fluorophores, to different fluorophores, and asked, do we tend to see single-labeled or dual-labeled neurons? And this is showing the OFC dorsal striatum projectors, ventral striatum projectors, and overlap. And we basically don't see any neurons that project to both places. So these are distinct, non-overlapping populations of neurons that are targeting the OFC. Okay. So if we were to perturb them, what is their effect on behavior? So Maggie used a viral approach to express this light-sensitive opsin, channelrhodopsin, specifically in the orbitofrontal neurons that project to the dorsal striatum. And if she stimulates them at the time of reward, what she finds is this interesting effect where she basically reduces the wait times. And this is significant, it's very apparent at the single rat level as well as at the aggregate. But one of the things that was striking about this is that the effect seems to be graded and have a graded dependence on the block, where the effect size is larger in the low blocks, intermediate in the mixed blocks, and weakest in the high blocks. And interestingly, she did not recapitulate that effect if she did the same experiment in the OFC neurons that project to the ventral striatum. So this seems to be reflecting something specific about the projections to the dorsal striatum. Okay, so again, we wanted to use our model to try to generate a hypothesis about what was going on. And what Maggie found is that if she introduced a bias to the prior, such that the model was biased to think that it was in a high block at the expense of thinking it was in a low or a mixed block, she was able to reproduce this graded effect of the manipulation as a function of block. And so that was an interesting hypothesis. We had difficulty finding any other parameters that produce this pattern of results, but it's a little bit of a weird thing to think about, that you're making the rat think it's in a high block. So we wanted to have kind of another prediction that we could test. So Maggie simulated the behavior of the model in a number of different ways, and what she found is that it makes a really kind of strange prediction about how wait times should change, specifically as rats transition from mixed into low blocks. And that specifically we expect a delay at this particular transition, but not really at the other types of transitions. So that's something we can test. So Maggie compared how the wait times change at the different transition types. And I'm just going to show you this transition from mixed to low blocks, but she basically found what the model predicted, which is that there's a specific delay in how quickly the rat's wait times change at transitions into low blocks in particular. And we didn't find any significant changes at the other block transitions, which is a specific prediction of the model, okay? So based on that, we do think that our current interpretation, at least, is that this manipulation is biasing rats' beliefs so that they think they're in a high block. But that's kind of weird, right? So why is that happening? So what we wanted to do was record specifically from these OFC neurons that project to the dorsal striatum and see what their task-related dynamics were. So to do this, Maggie decided to do a fairly heroic experiment where she's doing optogenetic tagging and collision testing. So just to explain what collision testing is, so again, she's using a viral strategy to express channelrhodopsin in these projection neurons. She's then implanting a neuropixel probe in the OFC and the fiber optic in the striatum, okay? And so what happens is she's going to shine light in the striatum, and that's going to elicit what are called antidromic action potentials that are generated at the axon terminal and then back propagate to the cell body in the OFC. and she can record those antidromic spikes with her neuropixel probe, and that's shown here, where the gray bar is the illumination period in the striatum. But every once in a while, these neurons are also going to generate orthodromic action potentials that originate at the cell body. So what happens is those orthodromic spikes, if they happen at a short latency relative to the antidromic spike, they propagate down the axon, but then the antidromic spike propagates up the axon, and eventually they collide. And because sodium channels have a refractory period, at those collisions, sodium channels on either side are inactivated, and so the current has nowhere to go, and your antidromic spike fails. Okay, so that's what's shown here. You have short latency orthodromic spikes that cancel your antidromic spike. And again, if you go far enough back in time, the orthodromic spike has time to propagate all the way down, and so it doesn't interfere with your antidromic spike. So these are very hard experiments, but it's really the gold standard for establishing that these are direct projectors. And we can do this for OFC neurons that project to the dorsal striatum, where we see collisions that then recover if you're far enough back in time. We could also do this for OFC neurons that project to the VS. And we think that it's really important to do this very challenging technique because cortex is strongly recurrently connected. And so if you're just using short latency low jitter spikes, you will find neurons that look like they're projectors, but they're failing collisions because they're network effects, okay? And so Maggie, over the course of a couple years, has recorded from 73 projectors that go to the dorsal striatum and 54 that go to the ventral striatum. And I just want to emphasize these are very low-yield experiments. Each of these rats takes months to train, and she gets 0 to 3 neurons per rat. And so we wouldn't be able to do this experiment without the high-throughput facility. Okay, so what do the neurons do? So what Maggie found is that the neurons that project to the dorsal striatum strongly encode the reward volume specifically at the time of reward. So this is a neuron that has a higher firing rate for large reward offers and lower firing rate for small rewards. Here's another one. So there's heterogeneity in how the rewards are encoded, where some neurons have positive value encoding, which is what we would call this, and others have negative value encoding. To summarize, over 73 neurons, we compute the D prime, which is basically how different are the firing rates on the high volume versus low volume trials. And we're doing this in an unsigned way, so we're basically allowing both of these guys to have similar D prime values in principle. And what we find is that the DS projectors care more about volume than both the VS projectors in purple and the overall OFC population. And yet, they don't explicitly seem to represent block. So this is looking at block D prime conditioned on 20 microliter trials only, okay? So it seems that to the extent that these neurons are updating beliefs, they're doing so as a function of their representation of the reward. But one thing that still kind of is very confusing about this is if we look at the signed D prime for each of the neurons, there's an equal fraction of neurons that encode positive value versus negative value. And so it's not obvious why optogenetically driving this heterogeneous population would bias your belief in a particular way towards a high block as opposed to the others. And so we're still trying to figure out what the mechanism is of this optogenetic effect, but I'm going to tell you just about one line of inquiry that I think is very promising, and that is using RNNs. So this is the work of a very talented postdoc, David Hawker, who is also co-advised by Christina Savin. And actually last week, his paper came out, which develops kind of a new compositional pre-training procedure for training RNNs that more efficiently produces animal-like behavioral strategies in our task, and also provides a normative approach for identifying pre-training tasks based on the formulation of the task as a Markov decision process, or MDP. But anyway, David had developed a way to train RNNs to perform our task. So just to briefly tell you, he's using deep meta-reinforcement learning agents with a modular architecture. So for most of the paper, we used LSTMs, although he's also replicated this in GRUs. And so you basically have one network that's performing inference. We think of this as putative OFC homolog, and it's setting the probability of each of the blocks to a second network, which is basically the policy head. And we think of that putatively like a striatum homolog. And if he uses his compositional pre-training approach, he can generate behavior of the RNNs that is qualitatively similar to what we see with the rats. And I should say it was highly non-trivial because this task has very many salient suboptimal solutions, it actually took a lot of work to train them to produce rat-like behavior. Okay. Yes, I'm almost done. So one of the things that is interesting and kind of unexpected is that when David simulates the optogenetic effect in his modular RNN, he actually reproduces what we see with the rat optogenetic result, which is not just a reduction in wait times, but with a graded dependency on the block, such that there's the greatest reduction in a low block, intermediate and mixed, and weakest in a high block. And one of the other things that's quite interesting about this is that this is also highly non-trivial. So whether or not he can recapitulate this result depends on the architecture. So we don't see this in GRUs, we only see this in LSTMs, which suggests that perhaps the saturating nonlinearity is an important component of capturing this result. And it's still, we're still kind of trying to figure out under what conditions this holds and doesn't, but it also is sensitive to the curriculum that the network was trained in. And in his paper, David found that there's, similar to this idea of degeneracy, right? There are different curriculum that can produce what looks like similar wait times, but if you look inside, they're using different dynamical motifs to solve the task, and we think that that is an additional thing that determines whether or not we can produce this optogenetic result. So what David is currently doing is trying to characterize in the networks that do reproduce this versus those that don't, what are the differences in terms of the dynamical consequences of the optogenetic perturbation? And then we're using that to guide our ongoing experiments in rats. Okay, so to summarize, I've shown you that OFC updates beliefs for state inference. OFC neurons reflect inferred state transitions in experts but not naive rats. OFC projections to the dorsal but not the ventral striatum bias rats' beliefs about hidden states. And we're currently using RNNs to recapitulate the optogenetic result, and we're trying to use what we learned from those experiments to guide our ongoing experiments in vivo. And with that, I'd just like to thank my lab and acknowledge the people who did the work, and thank you again for your attention.